All right. Hello, everyone. Thank you for joining today's Econ webinar. My name is Diane Bosanchuk. I'm going to be your technical support this afternoon. If you have any issues, any questions or any technical issues, please use the chat feature in the Zoom platform and chat me or any of the other of the panelists, and we will go ahead and I'll forward it to you if you have any issues. If you have any questions, we will have a Q&A session. If you have any questions, please use the Q&A feature and submit your question. That way we will then be having the session at the end of the webinar. For archival purposes, this webinar will be recorded and we will be posting it on the Beacon website later today, so you can watch it again. So I'm going to go ahead and hand this over to Anna Monfils, and she will go ahead and get started on the webinar. Thank you, everyone, for joining.
All right, thank you very much. First, in starting on this first slide, I want to make sure that I give credit to a lot of people who've done a lot of work in the collections and education world, and I'm just lucky enough today to get to present some of that. I'm also here somewhat representing the Biodiversity Collections Network that's charged with several tasks. I'll talk a little bit about that. But really we're talking about biodiversity collections and education and things such as workforce development, as well as the utility of collections in your standard undergraduate classroom or K through 12 classroom. I'm trying to, there we go. So there's sort of a history to the efforts to digitize about 1 billion US specimens that are housed in the nation's biodiversity collections. And it started with really a grassroots effort where a lot of collections and collections manager people got together to talk about sort of how they could preserve the collections and start digitizing the data. And out of that came an advisory document and a decision on the part of the NSF to form a program called Advancing the Digitization of Biological Collections, or ADBC. And ADBC had a certain amount of money that they used to fund a lot of collections that were clustered into thematic collection networks. And they also funded a hub, and that was Integrated Digitized Bio Collections, or iDigBio. And the job of the hub was to integrate resources and standardize best practices for the national digitization effort. Shortly after iDigBio was funded, within I think about a year, Beacon was funded as an RCN, and that's a research coordination network. And it was funded by NSF to support the development of a new and sustainable community of practice for the US biodiversity collections. And the goal was to build a community to advance and sustain the digitization. And this initiative was funded for 3 groups, which was AIBS, which is hosting this, the Society for the Preservation of Natural History Collections, and the Natural Science Collection Alliance. And this slide here is in real time today. It shows the number of specimen records that have made it onto the national portal at iDigBio. You can see that in your upper right of the screen. But what you also see here are all of the collections involved across the US. And if you were to expand this out, you see quite a few of those are on top of each other, and that there are quite a few collections involved. Now, with all this digitization that's been going on, we're starting to get more and more data and be able to ask research questions that are broadly interesting. And as a result of that, through the efforts again of iDigBio and several people at iDigBio, there's been, now we've had our 2nd year of the Digital Data and Biodiversity Research Conference. And this is a conference where people come who are using digitized data and talk about some of the different projects that they've been able to do. This year's was particularly interesting as we learned about a lot of different research that's being done, everything from looking at disease transmission to looking at epigenomics and the effect on disease, looking at zoonotic disease transmission, looking at some really interesting data that came out of NASA where they were able to discover a new colony of penguins. There's other interesting projects looking at parasites and parasite coevolution. And then there's, of course, interesting stuff that relates to more of an applied field, such as conservation of rare species, or even work that might work with invasive species. So lots of really interesting research that's coming out of this. Now, when the NEBA came together, they came together with an implementation plan. And Beacon right now is revisiting that implementation plan to look at what has worked or progressed, what needs to be revised, looking to the future. And really what I want to focus on here are what's going on with education. And there's 3 places where this really comes out in these sort of 6 major points of the implementation plan. One is enhanced training, creating the next generation of biodiversity informatics managers. That's clearly going to be workforce training and development there. But I think it also comes in when we talk about increased buy-in and participation from a broader range of stakeholders. We're going to have to educate a much broader swath of the public in order for them to understand the utility and buy into the utility of the collections. And then last on here, but I definitely don't think is least, is infuse specimen-based learning and exploration into both formal and informal science education. And that has taken on many different dimensions, from citizen science to outreach, to work in museums with specimens, to virtual work, to in the classroom, where there are current initiatives in K through 12, in the undergraduate curriculum for both majors and non-majors, and where there are emerging graduate programs that are dealing specifically with biodiversity data. So I like this figure because it really kind of shows you how you have the majority of the students that you're going to encounter in the K through 12 environment. Many of them go on to undergrad, a few will go on to grad, and a very few go on to become biodiversity scientists. So to really sustain our efforts in biodiversity science, we need to broadly train the developing workforce, harness the energy and excitement in the next generation of scientists, and explore novel uses to engage the audience. We basically need to harness that awesome energy the next generation of scientists is coming in with, and we need to help them develop the necessary data skills and the databases themselves that can be used to solve the big questions that the 21st century scientist is going to encounter. Now, the 21st century biodiversity scientist is going to have skill sets that borrow heavily from lots of different fields, including evolutionary biology, systematics, ecology, but also geology, environmental science. And they're going to have skills in quantitative, computational, and data type sciences, and be able to conduct research using large and complex data sets. So we're piling on a lot more skill sets. And there is a potential here for collections, both physical and digital, to really be a key part of training that next generation. So I found this slide online and liked it because I think that this is the reality of we are living in a rapidly changing world. We have a rapid increase in technology. People are far more mobile. There's a lot more connectivity. There's an increased level of diversity. Resource needs are higher, and there is an explosion of big data. And it's creating a different sort of landscape, both for the students going on to become part of the workforce and for the educators trying to teach those students. So this is a quote I see often, which data is the new science. The breadth of the data that I deal with now is completely different from what I dealt with as a student. And the skill sets that I have to have, and I now have to teach as a fundamental part of the undergraduate curriculum, have changed. So we need to teach students how to find data, comprehend it, interpret it, ask questions with fidelity to the data, and use the data appropriately. And that's no longer an optional set of skills, but a fundamental part of sort of undergraduate training. We also know that we are seeing an increased need for training, expertise, and innovation to address critical scientific issues of both national and global importance, things like climate change, zoonotic disease, resource management, invasive species, and biodiversity loss. And the rapid mobilization of data has increased our capacity to investigate these large-scale issues, and it's of critical importance that we train the next generation in how to work with that. We also know that we have a changing demographic and a highly diverse millennial workforce. Now, this is a positive in the fact that we know increased diversity in a working team is going to lead to increased creativity and productivity. But it provides a challenge in creating the materials that are both inclusive and culturally relevant to the changing demographic of the millennial workforce. We know too that there are far more 21st century skills And they put these in a category sometimes of called soft skills. Skills that we now require of people entering the workforce that are somewhat different from sort of the raw science skills that we're used to training our students in. So looking here, you see things like critical thinking. And critical thinking involves a huge component that looks into information discovery and interpretation and analysis, the ability to reason, construct arguments, and a lot of systems level thinking on a much bigger scale with lots of moving parts. Communication now more than ever is important, and it's important because it also has the digital aspect to it. So still in there with all this going on is the concept of effective listening, but also delivering oral presentations and communicating, such as venues like this, via digital media, engaging in conversation and discussion that's inclusive, and communicating in a really diverse environment, whether it be online, whether it be with collaborators all over the world. And that doesn't change the value of writing, and the value of writing to inform, support arguments, and engage. But there's an element there that is interesting. It's also there to entertain. And this idea that we have a lot more venues and ways to communicate to a much broader audience, and it's the expectation that this next workforce can do that. Collaboration is also an enormous part of what we're looking at in the future. And data has, or science has changed from being siloed to really being about shared and open access and collaboration, which is a different approach than we might have seen 10 to 20 years ago. We also see an emphasis more on leadership, but also on some of the skills like cooperation, flexibility, reliability, and how to be responsive. All things that can get different levels of challenge as we bring technology into them as well. And then the last one's creativity, and creativity is the hardest thing to train into people, but the freedom to create original ideas is going to be pivotal for us as scientists. So that's idea generation. That's design, refinement, being able to work creatively and not just alone, but working with others. So I would argue that there's an incredible potential in natural history collections to address these needs for the 21st century scientists. So natural history collections are going to be vouchered specimens, specimens housed in natural history collections with associated metadata that are archived in perpetuity. But these are things that are inherently engaging. They're concrete. Students can actually handle them. They can go back to that original data source in a physical form and ask more questions so they can practice the iterative process of science. And it can be place-based. You can actually look at what someone may have collected in your hometown, or where your grandparents live, or where your community exists. So some examples of really cool ones here is, I don't want to take credit for anyone's work, so I'll do my best to credit people. You'll also see that I try to give you links to where you can find these resources. When I point these resources out, if you have to go back later, you can always contact Molly, Phillips, or myself, and we'd be happy to direct you to where these are. This one was developed by Gary Motz, and it looks at, and it's really cool, it uses 3D printing to make prehistoric animal teeth and has those 3D printed teeth in class for students to look at and think about what prehistoric animals may have eaten, what their diets might have been like, and what the ecosystem they were in was like. It's really interesting, and it's a way to bring together some sort of fossil evidence and 3D printing. Here's another one that was published by Eileen Lacey, and this was a product of the AIM Up RCN-UBE. You'll hear me talk about that in a minute. This uses data from the Grinnell study in Yosemite and looks at 2 species of chipmunks and how their distribution has changed relative to sort of changing climate variables. And this is a module that's actually gone to publication, as you'll see here, but it looks at some really interesting stuff. People really like chipmunks, and it's really neat to have it working with the Grinnell study. Now, another thing about collections and their sort of unique attributes is that a lot of these newer sort of suggestions or of how we're to teach, which is more student-centered, is facilitated a little bit more easily using collections and collection-based data. They are ripe for incorporating collaborative learning activities, encouraging community participation, so reaching out to the community about different bits of data, creating societally relevant case studies, and looking at science in action sort of activities. So a couple examples of those. Here's one that was developed by a core team within Blue. We wanted to teach some pretty standard content that you'll find in introductory biology, pollination biology. And we wanted to do some basic tenets of coevolution, all falling under the normal content covered in that course. But we wanted to use it using specimen-based data. And so we went into the iDigBio website. We were able to pull up information on distribution of agave and of the bats that pollinate them, start looking at specialists versus generalists, impacts of shifts in when there might be a migration event. And then at the end, we look at vampire bats, which in some places coexist, and there can be some impacts of the control of vampire bats on the sort of more endangered bats that pollinate the agave. So this is one example. Another one, we just did this here at CMU, we're in our second iteration of it, is we're always looking for ways to get our undergraduates doing course-based undergraduate research experience. We have about 400 to 800 undergrads, and that's a lot. That would be a lot of undergraduate mentoring through individualized research projects. And so we put one into our core curriculum. Now I can only show you part of these because of the nature of what they are, but the students had to create infographics around a question or an idea, and they had to find the data, use the data, download the data, clean the data, use the data appropriately as part of describing something related to whatever their question was. And the question and the data sources went back and forth till they ultimately come up with these infographics, which was their final project. And so one group decided to look at Asian carp. They got really creative with their graphics. I wish I could show you through the whole thing. But they were able to find data like how much money is spent on each different sort of invasive species in the Great Lakes. They were able to look at how much was spent specifically on Asian carp versus other types of conservation initiatives. And we're able to break all of that down, finding original data to do it. The other one here was a bit of fun because they went and looked at plants they would like to not encounter while camping and looked up the distribution within the state of those plants and determined where they wouldn't want to go and where they would want to go based on the presence of organisms they didn't want to encounter. So the questions that they can ask can be fairly creative, but the idea is they go get the data, they ask the questions, and they look at the data and find the data that supports what they're saying. So another thing is the data is very experiential, it's verifiable, and it can be relevant to students. And so what happens with the data is we know that students separate from big data. So they do one of two things. If they gather data that gets contributed to a larger data source, they will give over the autonomy of their own data to the bigger data source. So my data wasn't valuable, but the big data source is. Or they do the inverse, which is the big data can't be real science since I contributed to it. So there's this disconnect with the data and the understanding of the data. And so what we can do when we have collection-based data is they can handle, they can touch, they can have sort of almost a relationship with whatever it is that data point that allows them to think all the way through the data continuum. And so they can start anywhere in the data life cycle, but even at the beginning of the data life cycle, all the way through generating, curating, finding, evaluating, and then ultimately using the data to address a question. So one of the projects that's done that pretty effectively is collectionseducation.org, where they've taken the standard, almost every plant systematics class will go out and do a plant collection. But what they have the students do, is we have them load their data up onto iNaturalist and work within the citizen science community and look at the process from gathering data all the way until it makes it into the overall database that shows the distribution of whatever those plants might be. And we've noticed over time, as we might teach classes a couple of times, the students start finding the data other students gathered and really connecting with what classes before have seen. But because we bring in that sort of citizen science component to it, and because we're going through the whole data pipeline, more like we do now in curated collections, students are having a connection to the data in a different way, and we're seeing a marked increase in their understanding of the data pipeline and of the role of citizen science in general. Here's another great initiative. I encourage you all to look this up. The next event is October 18th through the 21st. And this is a iDigBio digitization event or transcription event, where at many different schools all over, I believe, the world, at the same period of time, October 18th through 21st, there are mass digitization efforts. And there is a lot of coordinated activity. There is the ability to interact with the other teams. There's ongoing markers and numbers on the progress that people are making. This has been wildly successful and has gotten international and much bigger every year that it's done. And any group, anywhere can participate. You don't need your own collection to do so. I encourage you to reach out to this group if you're interested, as the next one's October 18th through 21st. And then last, and I think this is critical, is we don't have any more room in the curriculum for new content. And here I'm talking about all these skill sets now that the next year, the upcoming workforce will need. So we have to layer those skill sets onto content that's already there. What's nice about natural history collections is that the data that we can get and the specimens we work with align very well with the next generation science standards and the vision and change recommendations. And so the parity there allows for the extra level of adding real data and some real skills into content that's already being introduced in those courses. So taking a tiny step back, I want to give some credit to some groups that came before and did a lot of the sort of preliminary work to support the value of education with natural history collections. So one of the first RCNs that was funded, again, that's a research coordination network, was Collections Web. And Collections Web ran a special session on education. And alongside making the recommendation that we have a centralized data portal, which has been now done with the iDigBio Hub, they also talked about investing in the importance of investing in tools for instructors and students to access and analyze and visualize specimen data. It's important to incorporate educational experts into the process of generating collection-based materials, and how we need to further engage and continuing to engage teachers and museums across the US. As Collection Web was completing, the National Science Foundation funded AIM-UP, Advancing the Integration of Museums into Undergraduate Programs. This was an RCN UBE, so that's a research coordination network about undergraduate biology education. This it was funded for 5 years, they extended for a year, and over that period of time they had annual meetings and several small meetings to look at what it would take to get more specimen-based education in the classroom and how to get natural history collections into the classroom. When AIM-UP started, the digitization effort was just really getting some traction. By the end of AIM-UP, digitized data was much more of a usable content. When AIM-UP ended, we didn't want to see sort of the educational initiatives associated with natural history collections sort of stop. And so at that point, we did a survey of the community to find out really what it was the community felt we needed. So we did an assessment, and that was done in combination with AIM-UP, the Biodiversity Collections Network, and iDigBio. And from that, we had sort of several conclusions. The community wanted to see an integration of various efforts and centralized resources so that you could effectively and efficiently get to modules that may have been developed. There was a strong recommendation to incorporate biology educators from the very start. There was a request for developing meaningful undergraduate research experiences that would incorporate specimen curation and the data lifecycle. They wanted best practices that related to mentoring student workers in natural history collections. With the money that went into digitization, a lot of that went to undergraduate researchers or undergraduate students who were facilitating digitization. And the community wanted to know how to more effectively engage those students while they were there doing that task. There was a request to develop and refine educational modules involving natural history collection data. Molly and I were just talking about how just 2 years ago we were having trouble finding modules, and now we're finally getting some critical mass of modules that integrate and utilize the data. And then the last step was disseminating and propagating materials. So to disseminate and propagate really means more than just to publish or to develop for yourself. but how are you getting them out there and implemented by others? And so one way is to introduce modules in educational venues. Another is to incorporate partners who might be effectively doing that already. And a big one was if we could get it into the textbooks and the online resources, we might start seeing that show up in the classrooms more. But one of the biggest needs was a train the teachers sort of experience, something that would help the teachers become more comfortable with the content and the material. And this actually aligned pretty well with the recommendations of Vision and Change. And here's a quote from there, but it really talks about the importance of working on things in a group, the peer-to-peer mentoring, the ability to troubleshoot and work with materials, but do that sort of as a cohort versus in isolation. And this is one of the prime strategies that we've since adopted to help teachers not only become familiar and comfortable with the modules, but to help them adapt the modules for their own course, and then as a group mentor each other through the process of implementation. So after we got together and saw what these needs were, the next step was to try to pull the community together a little to address that sort of first and second request, which is centralizing resources. We also looked at this very real need to train students in data science and the sort of value of collections sort of across the curriculum and introducing students to data in a new and meaningful way. And we wrote a grant proposal and had an RCN funded. The RCN UBE is BLUE, Biodiversity, Literacy, and Undergraduate Education. This is a network or a community. It is not a couple individuals. It is meant to be something that the community can utilize and refine, and a place the community can go to get the resources that they need. Right now, we have some educational materials up and available. You can see them. We are rapidly adding material as we move along, as BLUE is in its first year still, and so we are still refining our methods of delivery, but hopefully anyone out there, you may have seen one of our talks before, but we are open to involvement or anything that advances the idea of getting specimens into the undergraduate classroom. iDigBio has been working at this for quite a while, and I have to give a lot of credit to Molly Phillips for being an excellent, excellent collaborator and builder of resources. So she has been sort of keeping the community together on the K through 12, as well as the undergraduate curriculum items, also managing a lot of the outreach. And what Molly has done is actually create a resource page. Now, I'm showing you here the wiki. There's a lot of information on this wiki, so I'll just quickly tell you. iDigBio has funded or co-funded several different workshops to train teachers. They are currently running webinars and have run webinars for the last several years, some in conjunction with AMOP, some in conjunction with BLUE, on the topics of education, on the topics of broadening participation, on the topics of outreach. And we are just getting into a new year of webinars, which we hope to have include some on broadening participation. But I think one of the many valuable resources would be the resource section. And I don't have a blow up of that because there's so much there. So what Molly has done is aggregate the resources that she has found or she has seen developed. And if you go to the AMOP website, if you go to the wiki, you go to the site, you'll be able to find those resources and click on them and get to the direct sources. You can get to BLUE from there. You can get to other initiatives such as the Geo community initiatives like CIRC and Integrate and other projects such as that. You can also find libraries of life and other sorts of materials. So it is an excellent resource, and we hope to sort of mirror some of that on the BLUE site as well. The other initiative that has also sort of come about in the last 3 or 4 years has been this QUBEShub. And the QUBEShub is about building community. So it's that same idea of creating the community around math and biology education to share resources and methods for preparing students to work with data of some sort in some way. And so this aligns very well with everything that BLUE and iDigBio have been working towards. And the QUBEShub provides some infrastructure for creating these virtual communities to work on materials. And so along with that, and this has been something that's just come down the pipeline I'm super excited about, is this concept of open education resources. So BLUE has an interface on the QUBEShub, but anyone can go to the site you can load up, and they have a sort of framework for doing this. If you have a module, if your module is 5 minutes or it's a 3-hour lab, there's a format to load that up, and once you load it up on that site, you will be able to monitor, and you can see here, you'll be able to monitor views, downloads. You will also get a DOI number so that you can track that. And if anyone was to come in and use your module, one, you'd see that they downloaded it, but if they were to revise it, they could actually link their revision to yours and you could track their numbers as well. They have assured me that by the beginning of next semester, they will be able to mask some of the content. So one of the issues we've run into with materials is I, as an educator, I want the answers to the questions because I want to make sure I'm interpreting the questions correctly. And I also want to make sure I know the answers to the questions. And if we put those out there, our students will find them, and it makes some of our efforts sort of moot at that point. And so this has been an ongoing concern, but this is something that they will deal with so that you can only have, instructors will have access to certain material, but students will not, in the event that you don't want them exposed to those answers. When AIM-UP ended, Blue, QUBES, and iDigBio, as well as many other partners that you see up there at the top, got together and sort of piloted one of these faculty mentoring networks. Now, we managed to take a very big bite instead of a small bite when we did this. We had 37 applicants, 21 ultimate participants. We broke our participants up into groups based on different modules, and we led different groups through this process. And then after the 2-day sort of in-person meeting where we kind of introduced materials and methods, had a lot of conversations about sort of what supplemental material might be needed, those groups went on to meet sometimes weekly, sometimes monthly, through the time that they implemented their modules. Now, when I was writing this up the other day, I realized it's a pretty significant thing that at the end of this, we had modules that were implemented at 16 separate institutions. And those are 16 institutions that didn't have digital data in their classroom of this sort before, and who now had it as part of their standard curriculum, which means those students who go through that curriculum are exposed to something they need, but isn't present in a lot of the curriculum out there yet. So this is exciting. It also gives us a proving ground for some of these modules. So, oh my God, I just am talking. So I want to talk about some upcoming opportunities. This is recorded, so all of this information will be retained. If you want to hit any of these sort of localities or tiny URLs we're going to give you. These are upcoming opportunities. Some are affiliated with Blue, some iDigBio, some others. We are not the only game in town, and we are happy to direct you wherever it goes, since I think ultimately our goal is to get those specimens in the classroom. Now, I haven't talked a great deal about this, but fundamental to sort of what we're doing with education and outreach with collections is this concept of inclusivity and broadening participation. And when we wrote the Blue original proposal, we sort of had to pull some items out of it. This was recommended that we pull out of it, but we've maintained it in other aspects. And that is to continue our efforts to broaden participation. So one thing we did do is we do have a grant that was initially funded by NSF to iDigBio to run these 2-day workshops in museums. And so you bring students to the museum, they can have a shadow day, and then they have a day where they learn about careers in biodiversity science, organismal biology. And it's largely volunteer. The funding pays only for the students to eat, so we make sure they eat very well. And we have found such an incredible value from the community of stepping forward to volunteer their time. And we have had a full impact workshop every time we've done this. So we have capped things at about 100 students. We've learned over time that we can cap at about 135 and get closer to 100. And we have had, in the case of the Field Museum, we had to turn away about half the applicants because there were so many applicants to come and participate. These are all undergraduate students. And then we bring in speakers, and we make every effort to bring in emerging professionals that can relate very well to these students and to sort of what the careers might be like from the perspective of someone who's not some, as I say, the old lady in the room. So we've done now a second iteration of these, which was funded. iDigBio passed this to the community picked it up, and there's several of us that are leading these. We will be having our second one in Flagstaff, Arizona in October. If you are in the area or are interested, we would love to speak with you. We always need help recruiting and need help when we have the students come in, giving them potential mentors. After that, we're looking for 2019 to look in Albuquerque and are in the process of working with the museum to establish that. Another thing that's coming up that's super exciting is the Life Discovery Conference, which is sponsored primarily by the Ecological Society of America, also the Botanical Society and the Evolution Society, or the Society of Evolution. And this is really a conference to disseminate and get materials for the classroom. And this was an incredible experience last year. I was able to go to it for the first time in Oklahoma. And this year, iDigBio is the main sponsor, and they're holding it in Gainesville, Florida. So that means this year, I sense there will be a heavy specimen-based component to this. And I strongly encourage you, if you have a module that you've developed, you don't even have to have it fully refined. Part of the process is having the group that you work with tell you ways that you could modify or improve or work on. You can run a workshop, or you can just run an informational session. You can also do these mini roundtables to talk about your products. This is a really positive experience, and this is a year where it's really going to be specimen-based, if I can say that, Molly. The other thing is we are looking at submitting for a share fair at SPNHC 2019. For anyone who went to SPNHC this year, we did a mini share fair in New Zealand, and we'd like to sort of ramp that up a bit when we're at the Field Museum to provide the option. Basically, this is like a round robin sort of scenario where you have people come in, they have a table, there's a period of time when people come sit at the table, go through the materials with whoever the instructor is or the developer is. They have some exchanges about it. You might leave with those materials if you want or provide feedback. And then after a time you shift and there's a whole new group of tables and new modules that are being sort of presented that you can go around and learn about as well. We think this will allow for us to disseminate things a little bit more and to help people with implementation. We are looking down the road to having a journal special issue of the TIEE journal. This is a peer-reviewed journal of educational materials, and it's teaching issues and experiments in ecology, but they will broaden out to evolution, and I think that we could argue almost everything we do falls under one of those. And you can basically publish in the TIEE journal. We're looking to develop some sort of framework. We're not 100% sure, but it might be a faculty mentoring network where people work together virtually to get their materials ready, review each other's materials, and ultimately submit them to the journal for the special issue. I'm excited to see this possibly move forward, as it would be a great way to centralize some awesome resources. So if you have any questions, please contact myself, contact Molly, contact any of the people that are listed on the front of this. There's many others that are involved in any of these projects. If you're interested in something you saw, just please feel free to ask either of us, and we'll connect you with whoever you want to meet with or connect with. There is a tiny URL. You can sign up to actually be on the listserv for Blue. You can also go to this tiny URL and tell us if there's something more you would want to do with Blue other than be on the listserv. So if you have a module, or if you want to be part of a faculty mentoring network, or you have some way that you think that you could contribute to the Blue community, or some idea to contribute, that would be where that you would put that. And then that's also the website. For iDigBio, ooh, looks like I forgot to capitalize there. There's the Education and Outreach Working Group, and to my understanding, and Molly can speak more on this, it is an inclusive group, so contact Molly if you're interested in being involved, and that is Molly's email address there. This is a group that talks about some really interesting and cool stuff. They meet periodically, but it's also a listserv there that you can get involved in and find out about any sort of workshops or events iDigBio might be doing. Another thing is, again, along the lines of broadening participation, that being a pretty fundamental aspect of what we do as educators, is we have a Moving the Needle initiative and a broadening participation working group. We had a webinar series. We are continuing the webinar series, but we're looking to more formally meet and start talking about where we could move and what directions we could move as a community to create a more inclusive and more diverse community of scientists. The last one I definitely encourage you to get a hold of is QUBES. You can get on, you do have to log in or create a login, but you have access to any of the materials on their site. You don't have to be affiliated with any one group or the other, though you could, if you wanted to, be affiliated with the blue group or all of the other. There's many, I think 140 some groups now on there. Then you'll learn about anything they're doing, learn about any faculty mentoring network type of stuff that might be sort of in process. That is also a very welcoming group. So just asking and reaching out to them, I think you'll get some positive responses. And then again, it's impossible to explain the amount of work that has been done by a lot of people to get us as a community to the point where we can digitize, to get us as a community even talking about the fact that we could use this data in the classroom. On here, you'll see most of the grants that have funded various parts of different things that I have spoken about. But I have to give a big shout out to Teresa Murad from the Ecological Society of America, who, whether she knows it or not, we've adopted her to sort of help us work through some of these different things, like the Share Fair or like the TIEE modules. And she has been very giving and with very open hand. So I think that's it now. If we have any questions or...
So yeah, if anybody has any questions, please use the Q&A feature and they will answer your questions.
And Molly, if there's anything I left out or said wrong, feel free to correct.
No, it all sounded great to me.
Because I talk about you to you.
Yeah, no worries. Sounded awesome.
We'll give everybody a couple minutes to go ahead and type in those questions. But again, just like Anna said, the webinar will be on the website later today. So if you want to go back and pull any of those resources or references that she mentioned, you'll be able to do that later today. And I will send out a reminder to everybody that registered how to access that.
I should point something out too. In putting the names on the grant awardees, I believe that there have been some changes in names since it was originally submitted, and it is now Rob Gropp, actually, that is the PI on Beacon. So I need to edit that and didn't see that till the last minute. I don't think we're going to get any questions.
I know, it looks like you just gave such an amazing webinar that nobody has any questions. You were so thorough.
Well, hopefully everyone feels comfortable approaching any of those names if they're interested in any of the products or projects, or have products of your own. We need more. We need to see what people are doing. I'm going to put us out of our misery.
Well, I was just making sure nobody had any questions again. And if you do have any questions after the fact, feel free to you can always shoot me an email at membership@aibs.org and I can point you in the right direction. So again, if your question comes to you maybe after the fact, you just go ahead and shoot it our way. Well, thank you everyone for joining today's webinar. Please visit the Beacon website. It's beacon.aibs.org to find out about any of the upcoming webinars that we have. We do have another one scheduled in October 17th. So again, more details are to come for that. But again, check out the website for more information. And again, thank you for joining everyone. You have a wonderful, oh wait, I think we have Oh, Molly says thanks. Okay. All right. Well, thank you everyone for joining, and have a wonderful rest of your week.
For joining.
Thank you.
Bye-bye.
Thank you. Bye.